So for this week's lecture, we're going to be talking about sleep and wake. We'll first tackle the rhythms that control sleep and wake, and then transition to talking about sleep in general and why we sleep, as well as the neurobiology of sleep function. It wasn't until the mid-20th century that scientists interested in behavior started exploring sleep. Prior to that, they thought that sleep just was, on the surface, associated with no behavior at all. But as I mentioned, around the 1950s, Eugene Arinsky started recording eye movements and brain waves from his 8-year-old son at the University of Chicago. And so he used his son as a subject to test a hypothesis about sleep not being a passive behavior, but rather something that involved a lot more activity. And it was at that point that we started to get an idea that sleep actually did involve behavior. Arinsky's advisor, who's Nathaniel Kleitman, shown here on the left, was the first scientist who really devoted his career to study sleep. He used himself, as you can see here, as a guinea pig. And here he is with another graduate student, Bruce Richardson, in Mammoth Cave, where he lived for about a month. He was interested in determining whether internal cues persisted even in the absence of external ones from things like light and night from the sun, and turns out that they did. He was also curious about the maximum amount of time people could endure sleep deprivation. And after staying awake for 180 consecutive hours, he determined that sleep deprivation could be used as a form of torture. I'm sure some of you have experienced the desperation that you have felt when you've really wanted to just go to bed. I know I have. So Arinsky, Nathaniel Kleitman's graduate student, wanted to explore eye movements for his dissertation. So was there a rhythm or pattern to these movements throughout the entire night's sleep? And turns out there was a rhythm. Arinsky found out that, ironically, when the eyes were moving, brain activity was similar to what it was when a person was awake. This stage is referred to as REM sleep, or rapid eye movement sleep. And when Arinsky's subjects were woken at this stage, they almost always reported that they were dreaming. And so what seemed to psychologists at the time as a passive brain state appears to have been just the opposite. A medical student that was assigned to Arinsky, William Dement, decided to pursue his interest in studying sleep and dreaming, and he wanted to characterize the different stages of sleep, as well as the patterns of activity that existed over the course of a night. What most psychologists thought was a very quiet time for the brain turned out to be quite the opposite. So most of our behavior demonstrates patterns, cyclical patterns, what you see here. So this period here is showing you roughly the time of 2 days. The nights are shaded in blue, and you can see that alertness changes in a cyclical pattern. So does temperature, rising the highest right before bed and being the lowest right when we wake up. Hormone secretion, such as growth hormone and cortisol, also show cyclical patterns. So there seems to be a rhythm to our physiology, a rhythm that lasts about a day. Circadian rhythms are rhythms that last about a day, circadian. An example of this that you do every day and can actually pay attention to is the sleep-wake cycle. And as I showed you earlier, many behaviors in and the physiology of animals show a circadian rhythm. For the most part, we use cues from the environment, such as the onset of light or onset of dark, and these cues from the environment entrain rhythm, rather control the timing of the rhythm. These cues are called Zeitgebers. In German, It's called time givers, literally. And if we removed the Zeitgebers and tested the sleep-wake cycles of humans, we would observe something that looks like this. So let me explain the chart to you, because if you've never seen one like this, it looks a little bizarre. So each of these dashes across represents a day. So we're looking over the course of 55 days or so, and these represent the number of hours. So it's showing you the amount of time people are kind of spending asleep. And what you can see is that they're sleeping for about 8-ish hours of the night. And as soon as we remove the Zeitgebers, which is around day 20, you'll see that the rhythm starts to shift, right? The time period when this individual sleeps shifts to the point where it doesn't follow a regular 24-hour day. So the entrained rhythm, the one that follows the Zeitgebers or the cues, lasts for about 24 hours. But the endogenous rhythm, one that comes from within, that's not using any cues from the environment, is called free-running rhythms. And it suggests that there's something within us that keeps track of time. What is this biological clock, and where can it be found? So scientists, neuroscientists early on, to figure out what brain regions do, what they did was they just destroyed brain regions and then looked to see if there were any changes in behavior. And so when they destroyed large areas of an area the medial portion of the hypothalamus, it disrupted the circadian rhythms and eating and drinking of rats. Interestingly enough, an area of the brain called the suprachiasmatic nucleus, or SCN, can be found in the medial hypothalamus. If you lesion the SCN specifically, you lose that free-running rhythm that you would see in the absence of Zeitgebers. There's no sleep-wake cycle that the animals show, no circadian rhythm, even when you try to entrain them with the Zeitgebers, right? Even when there's day and night, these animals show no rhythm. Just to show you an example of it, drinking activity in rats shows a circadian rhythm, largely because it occurs when they are awake. And so rats are nocturnal, just to remind you, and they tend to sleep during the day and are awake at night. So this is a type of activity tracking chart. This is the control animal that has its SCN, and this is the activity chart of an animal that is missing or has its SCN destroyed. So remember, in the dark, rats are more active compared to the daytime, and so you'll notice that each one of these dashes, right, each one of these represents a day. Each one of these dashes represents drinking, and you'll see more of them during the nighttime, which is when rats are most awake, compared to the light, right? During the daytime they're asleep, so they're not drinking as much. Maybe they get up a little because they're thirsty, but for the most part, most of their drinking happens in the dark. Look at the activity of drinking over the course of a day in a rat that has no SCN. There doesn't seem to be any pattern at all to their drinking activity. They drink about the same, but it happens at totally random times without any respect to day or night or any rhythm. To illustrate to you, the human beings also have an SCN, and here it is. So here's a coronal or frontal section through the human brain. This is the dorsal surface, this is the lateral surface, and this is the midline here. And what you'll see is that on the medial ventral surface of the hypothalamus, it's below this mustache-looking thing, this is all hypothalamus, the suprachiasmatic nucleus sits, right? So right here, on either side of the third ventricle, sits each nucleus of the suprachiasmatic nucleus, or SCN. It's called the suprachiasmatic nucleus. Let me erase my markings here so you can see it better. It is called the suprachiasmatic nucleus because it sits right above the optic chiasm, right? So if you recall from the vision lecture, the optic chiasm is the point at which visual information crosses, and the suprachiasmatic nucleus sits right above the optic chiasm. That's why it's given its name. So it's, again, not very creative, but you get an idea of where it's located simply by its name. So here's a suprachiasmatic nucleus of a rat. Each of these circles on either side represent the SCN, and here's the optic chiasm of the rat. It sits right above it, and this whole area here is the hypothalamus. So it's located in the medial ventral area of the hypothalamus. Incredibly, the SCN shows its own circadian rhythm. When you take it out and put it into a dish, it shows activity that lasts about a day. It has its own electrical activity, its own metabolic activity, as well as biochemical activity. So it seems to be keeping track of time, even when it's outside of the brain. But the most convincing evidence for the SCN being the internal biological clock came from this particular study. So let me set it up for you. Hamsters have a normal free-running rhythm that lasts about 25 hours. So remember, free-running rhythms are those rhythms that have no cues from the external environment. They are not entrained to any of the Zeitgebers. So they developed a hamster that they lesioned the SCN to, and so that hamster had no free-running rhythm, but they found a mutant hamster that had a free-running rhythm of 20 hours. So what they did in this experiment was they took the SCN from this mutant hamster, the one that had 20 hours, and transplanted it into a hamster who had a lesioned SCN and displayed no free-running rhythm. It restored the free-running rhythm of the hamster whose SCN was destroyed. But can you guess what the rhythm was? If you said the rhythm was 20 hours, you're right, because the only SCN, this hamster that had the SCN, its own SCN destroyed, was one that was 20 hours. So this illustrated to scientists definitively that the SCN was the internal biological clock. So the SCN controls free-running rhythm, but normally the structure is entrained, is controlled by Zeitgebers, and particularly those from the light and dark cycle. So how exactly does light control the SCN rhythm? How does it entrain the SCN? And this is how it does it. So information from the outside, light information, enters the eye just as it does when we are trying to see something. But there are specific retinal ganglion cells in the retina that actually respond to light, right? Prior to this point, I told you that light and dark, or light information, is only transduced by photoreceptor cells, rods and cones. Here's an exception to the rule. Here's a situation where there are retinal ganglion cells that are actually responding to light and dark, but they don't transmit any information about vision. All they're doing is transmitting information about light and dark, and they are part of the retinohypothalamic tract. This particular tract takes light information and sends it directly to the SCN. The SCN from this point then sends light and dark information to a gland that sits deep within the brain called the pineal gland. So in addition to controlling daily activity of sleep, wake, appetite, physiology, as well as temperature, the SCN sends light and dark information to the pineal gland. The pineal gland secretes melatonin. So I'm sure you've heard of melatonin because plenty of people especially these days, seem to be using melatonin to encourage sleep. And yes, melatonin seems to be a hormone that gets produced largely in the dark, right before we go to sleep. So melatonin seems to act as an internal Zeitgeber. It appears to be the chemical that signals to increase blood flow to distal areas of the body, which ultimately promote heat loss and starts the process of sleep. It seems that melatonin, rather than light, entrains circadian rhythms in individuals who are blind and incapable of getting light information from their eyes. So when melatonin secretion increases, right, close to the nighttime, when melatonin secretion increases, wakefulness is less likely to appear during the scheduled sleep phases of the day. So it seems that melatonin is considered a sleep facilitator and has been investigated as a potential treatment for insomnia. So essentially, because melatonin levels seem to naturally rise at night right before bedtime, and melatonin can serve as an internal Zeitgeber, an internal cue that it's supposed to be time when we're asleep, we can use melatonin as a way to cue the body when it's, in insomnia, when we're having trouble sleeping, cue to the body that it's time for bed. Melatonin, some research seems to indicate that melatonin can be used to induce what's called phase shifts or realign the circadian rhythm for those who might be suffering from something called jet lag. So for those of you who have ever traveled time zones or lived through daylight savings time recently, maybe not so much in the fall, but in the spring, you've experienced phase shifts. Phase shifts are a desynchronization of your circadian rhythm with the environment. They are out of sync. So the rhythm that you're in, right, the environment that you're in, is shifted or is different from your internal circadian rhythm. And when this happens, when there's a mismatch between your internal rhythm and the rhythm of the outside, and training cues, it can produce negative side effects such as difficulty going to sleep, waking up early, changes in appetite. All of these symptoms are part of a collection of symptoms known as jet lag. You can also experience it not just by traveling, but if you have shift work. People who engage in shift work have irregular schedules where they wake up typically when it's nighttime and are attempting to sleep when it's broad daylight. So there are, unfortunately, long-term consequences of shift work, right? The regular changes in the work schedule that result in us constantly having to readjust our circadian rhythm based on our work schedules can have some long-term side effects or long-term consequences, including health effects such as obesity, diabetes, as well as a greater risk of accidents. And so using over-the-counter melatonin as well as light therapy, so using light right before we wake up as a cue that it's time to wake up, can help with realigning biological rhythms with the environment and prevent these unnecessary consequences of things like shift work. So we're going to transition to talking about sleep for real. Rather than discussing the rhythms that control sleep, we're going to talk about the patterns that exist in sleep. So we can define sleep as a reversible, not irreversible, that would be bad, reversible state characterized by minimal movement, a relaxed posture, and diminished attention that's directed to the environment. There are a total of 4 stages of sleep, 1 through 4, 1 representing the lightest stage of sleep, and 4 representing the deepest stage of sleep. In fact, stages 3 and 4 can be combined to be called slow wave sleep, or SWS. We can further characterize stages of sleep into either REM sleep or non-REM sleep. Rapid eye movement sleep was the one I mentioned earlier in the lecture, where the, what's his name? I can't remember his name anymore. Oh, Arinsky's son demonstrated movements of the eye when he was sleeping, and when he was studying his subjects, he found that a lot of the times when he woke them up during this REM stage of sleep, that they were dreaming. All the other stages of sleep that are not REM are considered non-REM sleep. So stages 1 through 4 are considered non-REM sleep, whereas REM sleep kind of falls into a category on its own. It's not stage 1, 2, 3, or 4. You can measure the stages of sleep using something called a polysomnography or polysomnograph. They are a physiological recording technique that is using 3 different instruments. They're using an EEG, electroencephalogram. They're using an EOG, electrooculogram, so the eyes, and EMG, or electromyogram. So each of these are measuring a different aspect of the face, and here is a polysomnography. You're having electrodes stuck to the scalp to measure brain waves in EEG. You have electrodes attached to the eye to measure eye movement through EOG. And here you have electrodes attached to the muscles of the jaw to measure muscle tension during the various stages of sleep. So this shouldn't come as any surprise to you, but as we sleep, different stages of sleep appear to have different activity in the brain, in the eyes, as well as muscle control. And we'll talk about that later on in the lecture. But let's talk about what the EEG is measuring. So here is a blown up version of what we might be seeing under an EEG electrode. So the electrode is measuring brain activity on the surface of the brain. And so here's the skin, the skull, 3 layers of the meninges sitting right above the brain. And as you know, neurons use electrical activity to communicate, to transmit information. They have depolarizations and hyperpolarizations. So the neurons that happen to be on the surface of the cortex, generate so much electrical activity, particularly if they are active at the same time, that it's picked up by this electrode and then transmitted to an amplifier that makes the signal larger so we can see it. So when you read an EEG, the amplitude or the wave, the height of the wave of the EEG depends on the synchronicity of neurons. In other words, if neurons are activated at the same time, the height of the wave is going to be bigger. So simultaneous activity of groups of neurons creates a signal that's much, much larger and is recorded on the EEG. So a good analogy of this is having a group of people kind of randomly talk. It would just sound as chatter. But if you have a group of people calling out a chant at the same time, their voices would sound louder. So in the same way, when neuronal activity is synchronized, happening at the same time, the height of the EEG, the wave of the electroencephalogram, is much, much larger. So just to illustrate, right, here's the EEG picture I showed you earlier, and here are 6 different neurons that we're recording from. When the activity pattern of these 6 neurons is random or irregular, it generates an EEG that sums up to small waves. However, if the activity of these 6 neurons is more synchronized, that means they show waves that are activated at the same time, we would see the amplitude, right, the height of the waves being larger when we sum up all of their activity. So in this way, synchronized neuronal activity is represented as larger waveforms in the EEG. So there are many different rhythms that the brain shows, and they're called alpha, beta, theta, and delta, based on just what neuroscientists call them. So the second fastest waveform, don't ask me why it's the second fastest, it was probably the first to be discovered, which is why it's called alpha, but it seems to be the second fastest waveform. It's about 8 to 13 hertz, right? So the unit of measure for these waveforms is hertz, and so the number or the value of the waveforms of the second fastest waveform, known as alpha, is 18 to 13 hertz. It happens typically when we are quiet, maybe a little bit drowsy, but in the waking states. If we're zoning out and kind of daydreaming, we might show alpha rhythm activity in our brain. Beta is the fastest rhythm, and it's anything that represents 14 hertz or greater. It signals an activated cortex. So when we're really engaged and puzzled and thinking about something, our brain is going to show beta activity. Theta and delta are typically only seen during states of sleep. So theta lasts between 3 to 7 hertz, and delta, which is the hallmark of the deepest wave of sleep or slow wave sleep, is 4 hertz or 3 hertz or less. It is the largest amplitude, and you only see this waveform in stages 3 and 4 of sleep. So it is the slow wave sleep I mentioned earlier. Here are some examples of waves that you might see in the different stages of sleep, right? So rather than putting numbers to it, let me show you what these waves look like. So when we are awake and alert and really engaged in something, we would see kind of random activity of neurons leading to very low amplitude, but beta waves, right? And if we are sleepy and drowsy or daydreaming, we would see alpha waves. You can see that there's more synchronicity and the waveforms are getting bigger. Theta waves are low intensity slow waves. They are much, much slower than delta waves. So here is an example of theta waves seen typically in stage 1 and stage 2. Slow wave sleep demonstrates delta waves. Look at these massive waveforms seen during the slow wave sleep. You only find theta waves in stages 1 and 2, whereas these delta waves that I'm showing you here can be found in stages 3 and 4, which are combined to be called slow wave sleep. In stage 2 of sleep, you would also see waveforms such as sleep spindles and K complexes that are kind of seem random and happen at unexpected times. These two waveforms that happen in stage 2 of sleep are thought to play a role in things like learning and memory as well as sensory processing. You'll notice, if we look at the bottommost panel here, that the waveforms in REM sleep are very similar to what we would see when we are awake. It seems to be showing beta waves. When we are sound asleep, we show waveforms of when we are alert and engaged and awake. This is one of the reasons that REM sleep is known as paradoxical sleep. The reason why it's called paradoxical sleep, because we're asleep, and yet we are showing EEG activities that are similar to when we are the most alert when we are awake. So it makes no sense, right? It's a paradox. It's something that shouldn't happen and shouldn't occur, and yet it does. So as I mentioned earlier, Demet, a medical student who was working with Araski, set out to characterize the stages of sleep. So researchers have worked out the details of sleep stages throughout the course of a night, and this is kind of what it looks like on average, right? All of us are different, and depending on how much sleep we've gotten the night before and what we've been doing before we went to sleep, these stages might vary. But on average, this is the pattern of sleep that we experience over the course of a 7 to 8 hour night of sleep. You'll notice that we cycle through pretty much all of the stages of sleep. So when we're awake, it represents the most superficial areas. And then as we progress into stages of sleep, we get deeper and deeper into sleep, and this is time. And roughly around 70 to 90 minutes after we've fallen asleep, we see the first evidence of REM sleep, right? Remember, REM sleep is rapid eye movement sleep, and REM sleep is typically when it's thought that we dream. You'll notice that as we progress through the night, the slow wave sleep, right, shown here, gets more and more infrequent, right? When we get into 5 or 6 hours of sleep, the slow wave sleep completely disappears. So we tend to experience slow wave sleep earlier on in the night. Instead, what we see, unlike slow wave sleep, REM sleep seems to occur more frequent, and it lasts much longer than earlier in the night. So the period of time that REM sleep occurs exists for much, much longer than it does earlier in the night. And then we wake up. So let's contrast the differences between slow wave sleep and REM sleep, because in addition to happening more frequently or infrequently over the course of the night, they differ in very specific ways. So during slow wave sleep, most functions like heart rate and respiration is drastically reduced. It seems to be a period of rest for the rest of the body. REM sleep, on the other hand, the heart rate increases, blood pressure increases, and breathing gets more rapid and variable. During slow wave sleep, we have decreased brain activity, little to no eye movements, and maybe some muscle movements here and there. But during REM sleep, there's increased brain activity, right? There's that beta activity that I showed you earlier, and then there are eye movements that occur. But ironically, the rest of the body is completely paralyzed, right? There's no muscle movements because we don't want people to be acting out their dreams. So during REM sleep, the rest of the body demonstrates atonia, or loss of muscle tone. The only thing that's really moving is are the eyes. And we'll talk about what the functions of slow wave and REM sleep are in the second lecture. In slow wave sleep, it's very difficult to wake somebody up. It would take loud noises to wake them up, or a lot of shaking to get them up. And if they do wake up, they will act groggy and confused. And if you tried to test them on any cognitive test, they would have very impaired mental performance. On the other hand, with REM sleep, they won't react to noises, but they will arouse easily to meaningful stimuli, like their name being called, even quietly. And when they do wake up, they will seem alert and attentive and perform much better on cognitive tests than if they had just been woken up from slow wave sleep. When people are woken from slow wave sleep, they won't report that they're dreaming, which is the story-like progression of events. They might have some vague thought or emotion or image associated with something that was happening during slow wave sleep, but there's no story-like progression that is typical of dreams. On the other hand, during REM sleep, when they awaken, subjects almost always report that they're dreaming. And you see this story-like progression of events, like this happened, and then I moved here. And so REM sleep is typically when it is thought that dreams occur. Whereas slow wave sleep, even though the body is capable of moving, and it's during slow wave sleep that you have sleepwalking and sleep talking, the person's not really dreaming, not acting out their dreams, unlike what most people might think. It's slow wave sleep that we have muscle movements, because remember, in REM sleep, when we are dreaming, everything, the whole body except the eyes, are completely paralyzed. So here's a summary for this lecture. So the human and mammalian behavior seems to follow a circadian rhythm that lasts about 24 hours. The SCN of the hypothalamus controls free-running rhythms, but can be entrained using Zeitgebers, such as day and night cues from the environment. Light entrains the SCN via the retinohypothalamic tract, which also controls melatonin secretion from the pineal gland. Rhythms can get desynchronized from the environment in phase shifting and can lead to the symptoms that are commonly associated with jet lag. We sleep when melatonin levels are high and undergo 4 different stages of sleep, non-REM sleep, as well as we experience REM sleep. Brain waves get more synchronized, so the amplitude or the height of the wave gets higher with deeper stages of sleep. REM sleep is thought to be paradoxical sleep because the brain waves seen during REM sleep are similar to when we are the most alert and awake, beta waves. There are many differences between slow wave and REM sleep related to bodily functions, brain activity, response to external stimuli, as well as dreaming. So that's it for this lecture. Don't forget to submit your reflection.